Endometriosis is a complex and often misunderstood condition that affects millions of women worldwide. This condition manifests when tissue resembling the uterine lining begins to grow outside the uterus, leading not only to excruciating pain and inflammation, but, in some cases, also affecting fertility. Despite the widespread prevalence of this condition, the exact causes of endometriosis remain a mystery, with various theories considering genetic, hormonal, and immunological factors that need to be pieced together like puzzle pieces in a multifaceted picture. A new and exciting area of research is increasingly coming to light, shedding light on a previously overlooked connection: the relationship between endometriosis and the gut microbiome. Our gut, aptly referred to as the second brain, is not just an organ of digestion, but a highly complex ecosystem that houses trillions of bacteria living in a symbiotic relationship with us. In fact, we carry more of these foreign microorganisms within us than our own cells, challenging the notion that we are alone. These microbes significantly influence our well-being, regulate our digestive processes, and shape our immune defense, raising the question, could this microbial universe also play a key role in the development and progression of endometriosis? If so, we may be on the brink of a paradigm shift that could fundamentally change our understanding of the disease, akin to discovering a new continent in medical research. Recent studies suggest that this may indeed be the case. Research has shown significant differences in the gut microbiome of women with endometriosis, compared to healthy women, indicating that the composition of bacterial communities in the gut may influence the condition. A study published in 2024, which examined over 1,000 women, found that while overall bacterial diversity was not significantly different, certain bacterial species were associated with endometriosis. While some bacteria were more prevalent, others were significantly reduced in affected women, supporting the idea that the microbiome functions like an orchestra, where the absence or overabundance of certain instruments can disrupt harmony. The connection between endometriosis and gut bacteria becomes even more interesting when considering how metabolites produced by the gut, such as short-chain fatty acids, can affect the body. These compounds, acting as chemical messengers in the body, have implications for inflammatory processes and hormonal activity, particularly estrogen, a crucial factor in the progression of endometriosis. Studies suggest that some of these metabolites produced by gut bacteria could reduce inflammation and inhibit the growth of endometrial tissue, opening up potential therapeutic approaches that could act as a key to a locked room of treatment options. Furthermore, the concept of leaky gut is being explored in relation to endometriosis. A dysbiotic or imbalanced gut microbiome can increase intestinal permeability, facilitating the entry of toxins and pro-inflammatory molecules into the bloodstream, which could further exacerbate chronic inflammation in endometriosis. This increased permeability, acting like a leaky sieve, could intensify the symptoms of the condition and complicate treatment. While these findings open new perspectives for understanding and potentially treating endometriosis, it is important to consider the complexity of this condition. Not all studies agree on the significance of changes in the gut microbiome in endometriosis. Some researchers found no substantial differences in bacterial diversity, suggesting that certain species may play a role, but the entire microbiome may not be the primary trigger, fueling further discussion about the causes of the condition. In addition to observational studies, advanced genetic analyses, such as Mendelian randomization analysis, have identified specific bacterial genera that may be associated with different forms of endometriosis, suggesting a possible causal relationship. Although these results are promising, comprehensive studies are needed to fully understand the precise role of the microbiome and to unravel the complex interactions among the various factors contributing to the development of endometriosis. One of the most fascinating current research areas involves 4-hydroxyindole, a gut-derived metabolite that is found in lower amounts in women with endometriosis. Preliminary experiments suggest that supplementation with this metabolite could inhibit the formation of endometrial lesions and alleviate pain in animal models, making it a potential candidate for microbiome-based therapies. This discovery could be like a ray of light in the darkness of endometriosis research, opening new avenues for innovative treatments. In summary, the interactions between endometriosis and the gut microbiome represent a still young field of research that unfolds like an undiscovered land. We are just beginning to understand the possible influences of these microscopic organisms on the condition, and the prospect of new, non-invasive diagnostic markers, as well as innovative treatments based on microbiome research, appears promising. A deeper understanding of these connections could provide millions of women facing the challenges of endometriosis with better treatment options, nurturing the hope for a future where this condition is no longer seen as an insurmountable obstacle. That's it for today's episode. If you enjoyed our video, please subscribe to our channel and give us a thumbs up. We hope we were able to provide you with valuable insights and that we can continue to accompany you on your health journey. As always, we wish you a long, healthy, and happy life. Take good care of yourself and stay curious. Until next time.